I had another request to review this quiz on biological molecules. I'm not going to review all the questions because some of them, for example, if you don't know what a polymer is, I mean, this is an answer you can easily find in your textbook or in the lecture videos that I've already provided on biological molecules. It's in the very first molecule. There's an analogy to a pearl necklace. So maybe I'll just, identify where you can find the material for some of these, but others I will review. Okay, so polymer, that's something you can find by engaging with your textbook or the video lectures. Pearl necklace in the video lectures is a big old picture of it in the first lecture video for biological molecules, which is an introduction into biological molecules. All right, then we have dehydration reactions and hydrolytic reactions or hydrolysis, if you want to call it that. Okay, in the image below, A represents a blank reaction, B represents a blank reaction. All right, these are hydro whatever reactions because they've got water involved. You're either pushing water in or taking it out. So again, I'm not going to tell you the answer. I'm just going to give you information that might help you think about it so that you can remember it in the future. The thing is I really find that if I just give away all the answers, it actually doesn't help anybody learn them because that's the difference between active and passive study efforts. So remember, passive study efforts is when you're just reviewing material that you've already reviewed before. And I know that a lot of my students will review for hours and hours and hours and still fail the exam. The reason is that passive study activities are crap. I'm sorry to say it, but it's not that I think that people who use them are lazy. They work really hard for no return, which is terrible. So don't do it. You need to test yourself. And that means active engagement with the material. If you can't understand something, passive study methods won't tell you because you'll read it and you'll think to yourself, yeah, that makes sense to me. Well, does it though? Because if you can't answer these questions, then it clearly didn't. But you only found out that you couldn't answer questions by trying to answer questions. You didn't find it out by reading the textbook. So when you read the textbook, you thought, yeah, that generally makes sense to me. And then you might have come to the quiz and been like, I don't know how to answer this. Okay, so clearly reading the textbook didn't show you very clearly what you do and don't understand. So don't study by rereading it, okay? Unless you're actually looking, now the difference is if you're actually seeking the answer to a question, because that logs it in your mind, because you're actively searching, you're actively telling a story. So then stories we log in our mind. We don't just log background information. Our brains are set up to filter background information. So if you're studying by rereading and rereading and rereading something, that's just like putting it into the background filters. But if you're solving a mystery, and correcting mistakes and things like that, that logs the information differently. So I try not to give away too much because I think, for some of these, it's, these are things that you can search. So we talked about this in class. So hydro means water. It's really good in biology to remember the meaning of these prefixes and suffixes. They have Latin or Greek meanings, and you see them over and over again. And you'll get to a point, if you can remember what they mean, that you can just figure out what something is by its name alone. So hydro means water. When you hydrate, you're drinking water. So that's easy to remember. So here you can see H2O. You're removing it here. That's going to be dehydration. When you're dehydrated, you're losing water. So this is a dehydration reaction because water is lost. You had a couple hydroxyl groups here. You took away one of the oxygens and two hydrogens. It's a dehydration reaction. Please, if you don't recognize this reaction, watch the videos on the intro video to biological molecules. This is how all biological molecules are built, every single one. So you need to know, there's so many hydroxyl groups, so you really need to know what that is. It's dehydration. Okay, hydrolysis. What does hydrolysis mean? Or you can call it hydrolysis if you like. Hydro is water. Lysis means to break. And again, lysis will come up so much. You've seen it on your superbug activity. Again, I'm sorry we didn't do that in class today. I really regret that, but I can't do anything about it. So hydrolysis. That's when you break something with water by adding water. So that's that there. A product and substrate, we haven't really covered this material yet. We will get to this after exam 1. We'll talk about enzymes and chemical reactions, products, substrates. Substrates and reactants are the starting materials. So in each of these, this would be the substrate or the reactant. These would be the substrates or reactants. And then these would be the products. That's the end result. Okay. So not all of this is a substrate or a product, and not all of this is a substrate or a product, right? The starting material is substrate, the finished material is product. But we will cover that more in the future. Okay, question 3. Lysozyme is an enzyme in your tears, sweat, and saliva that destroys bacterial cells. If you denature the enzyme, why won't it work anymore? You need to understand what denaturation means. Denaturation is when you heat up a protein so much that it loses its shape. Okay, so if you denature, so you would have to look that up. So if you denature the enzyme, why won't it work anymore? If you change its shape by boiling it or putting it in acid, why won't it work anymore? Well, that's because an enzyme's form, any protein's form, is essential to its function. If you change its shape, it will no longer do its task. And if you don't know what the roles of proteins are, you need to watch the videos on the video I made on proteins. There's a long list of jobs that proteins have in the body. They are not energy sources unless you are in an extreme state of starvation. Our proteins have many other more important jobs than energy. So carbs, energy, yes. Fat, energy, yes. But protein, no. There's this long list of functions for proteins, and each of those functions really depends upon the three-dimensional shape of that protein. All right, so what types of subunits are DNA molecules made out of? Choose the one best answer. And I even said in here the full repeating subunit, not the smaller components. Okay, so you know that each DNA and RNA molecule is made up of repeating subunits. Part of that repeating subunit is the sugar phosphate backbone. Remember, sugar phosphate, sugar phosphate, sugar phosphate. Okay, so just with that information alone, phosphate by itself is not the repeating subunit. It's part of it, but it's not the whole thing. The 5-carbon sugar, part of the repeating subunit, but not the whole thing. And what's sticking off from those? Nitrogenous bases, that's not even listed here. But again, part of the repeating subunit, but not the whole thing. It's nucleic acids. You can also call them nucleotides. Nucleic acids include, the monomer component. A mononucleic acid includes a sugar, a phosphate group, and a nitrogenous base. So there are 3 components to each monomer in DNA and RNA. Okay. Then we have, which of the images below represents a hydrocarbon? Hydrogen and carbon is just hydrogen and carbon. So if there's other things in it, like here you have nitrogen, that's not a hydrocarbon. And you have phosphorus and oxygens and hydroxyl groups. By the way, this isn't a nucleic acid and a nucleotide. Okay. This is not a hydrocarbon because look at all these hydroxyl groups and oxygens and such. Well, does it have anything other than hydrogen and carbon? No. That's a hydrocarbon, but let's just make sure this one, no, that's got a nitrogen there, a carbon here, a couple oxygens. That's no hydrocarbon. These are all organic molecules because they have a carbon backbone, like the skeleton of these structures is mostly carbon with the hydrogens around them and everything, but they've got, all except C have a bunch of other things involved. Then you have, here's an amino acid. Which letter sits beside the amino group? So I'm just asking you to identify the amino group. I just sent out another video on that reviews the homework assignment, homework 3. And I had to identify the amino and carboxyl group there. So I will let you, all you have to do is look at the functional groups in your textbook to figure out which is the amino group, right? There's a couple, there's this functional group, there's this functional group, which is amino, which is carboxyl. And again, you can watch the protein video that I've posted online. I have videos for each of the different biological molecules that we've covered this week. And then I ask you to figure out which is the carboxyl group. So again, if you don't know, please watch my homework review and the protein review. Okay. Some people like videos more than they like textbooks. This information is also in your textbook, of course. But if that's like something that's confusing you, I'm not trying to be a jerk by withholding the information. I just, if you can't even find this information, please watch the video on proteins, please, because it means that you have no recollection of anything we did in class regarding this. Maybe you were absent, or maybe you just didn't understand. But this answer is explicitly in those videos and in the textbook. And I think that if you watch the video, I talk about the repeating subunits and the functional groups and what they are. And you need the whole, giving you the answer isn't going to help it stick. You can't memorize everything in biology. You just can't. I'm terrible at memorizing, but some people are really good at it. But the vast majority of you are not going to be memorizing your way through this. You need to understand it, which means, if knowing what an amino and a carboxyl group is like way outside of you need to review what the functional groups are in biological molecules and review proteins. I give a full discussion on what an amino acid is, and you need to know it from beginning to end. It's not enough to just try to memorize all the functional groups and what they do. You need to understand what they're for, Okay, so what is the description of each level of protein structure? Okay, again, this is in that video. Please watch it. You have proteins have 4 levels of structure, and like I just told you, the structural component of proteins is essential to their function. And there is a really important relationship between your DNA and protein, because DNA codes for protein. DNA does not code for carbs. It does not code for the phospholipid bilayer. It doesn't code for lipids or anything. It codes for protein. And proteins build all those other things. Okay, proteins build vitamins. Proteins build lipids. Proteins build hormones. Proteins build everything. So proteins are the robots that get everything done in the cell. They have many, many tasks. They're dynamic. They move, they will keep the cell alive. Everything else is just structural. Proteins can be structural, but a lot of them have these like the rules in defense and communication and chemical reactions and all the stuff that is in that video. So they have this complicated structure, and it's essential to what they do. So most of the time, proteins, to do their job, have to bind to something else. So for example, proteins whose job it is to communicate, they might be a communication signal, or they might be a receptor for a communication signal. And either way, okay, so if it is like, so some proteins are hormones, and that allows one part of your body to communicate with another part of your body. Like your brain could release a hormone and act on different tissues in your body, causing them to do different things. Okay, so if it's a hormone, that's a communication signal. And it's designed to perfectly fit into receptors for that signal. So the only tissues that are going to respond to that chemical signal are receptors that have a shape to fit, like a lock and key. So just like a key can only open doors, can only act upon doors that it fits, whose lock it fits, same thing. There's a very delicate and specific shape matching between the communication molecule and the receptor. So if either of them change shape, the signal can't be received. So one example, oxytocin. Oxytocin is known as the love hormone because it's very high in the blood of people who are feeling warm and loving towards others, and it causes them to behave altruistically and things like that. It's actually got more complicated roles than that. So it has this role as a neurotransmitter, changing our behavior and our feelings, but it also has a role on smooth muscle for women. Oxytocin acts upon uterine tissue to cause labor contractions during childbirth. And also, oxytocin acts on smooth muscle in the breast to cause milk ejection for the baby after birth. So men don't have, so oxytocin is filling the whole body, but it's not causing all muscle to contract. It's not even causing all smooth muscle to contract. It's only acting upon the smooth muscle of the uterus and of the milk ducts. Why? Because those are the only places that have receptors for that hormone. Now, if the shape, so those receptors are shaped perfectly to bind like a lock and key to oxytocin. If the receptor changes shape, oxytocin can't fit, and that tissue will no longer be able to respond to the signals of oxytocin. So you get a change, all of this, the receptors are a protein. All this is to say that a change in the three-dimensional shape of this protein is going to result in a change in functionality and maybe even a complete loss of functionality. DNA codes for protein. So this means that when we get to DNA and we're going to talk a lot about mutations and the effect of mutations on individual proteins. We've talked about mutations in terms of evolution already. But what I will want to talk about then is the mechanism. Why does a mutation change, coat color or hemoglobin or, a blood type? Why would mutations change these things? What exactly is happening? What is the mechanism? So you will learn at the end of this class, you'll learn exactly what a mutation is, exactly how they happen, and exactly what the effects are, the mechanism by which it affects the individual. And that will really feed into what we've already talked about in terms of evolution and natural selection and things like that. So this piece is really critical. I really want you to understand it. I really want you to watch the video on proteins to understand this. We covered it in class, but I know that sometimes you need, this is all very new. You need repetition. So I'm explaining the importance so that you know why to study it. Primary structure is the first level of structure. So what's going to be the most basic level of structures? So a protein is a sequence of amino acids, right? And there's 20 amino acids. So again, if you don't know that, then watch the video. So what's going to be the most basic level of structure? The sequence of amino acids in the chain, or two or more separate polypeptides held together by interactions between R groups, or complex folding patterns held in place by chemical interactions between R groups, or alpha helices and beta pleated sheets held together by hydrogen bonds between amino groups and carboxyl groups of non-adjacent amino acids. Well, the very basic level of structure is going to be just the sequence. So you have this long string of amino acids, and there's 20 possible amino acids. What sequence are they listed in? This has nothing to do with three-dimensional structure. This is just a list of what's in the string in order. Okay, that's going to be primary structure. Secondary structure it doesn't have anything to do with the R groups. It has to do just with hydrogen bonding between amino groups and carboxyl groups of non-adjacent amino acids. Tertiary structure and quaternary structure are both R groups. And again, the lower levels of structure are going to be more simple. So tertiary, the lower of the two, is going to be R groups within a single polypeptide chain, and quaternary structure is going to be more than one polypeptide chain held together by R group interactions. If that made absolutely no sense to you, I can't, there's already an entire video with videos, diagrams, drawings, for you to benefit you. So please go to that. It is in the biological molecule unit. It's called proteins. So you have access to that. And if there's something in it that doesn't make sense to you, let me know. But really, there's nothing I can draw here that will be better than that. All right, so 9. A mutation in a section of DNA causes a ribosome to place an incorrect amino acid in a protein chain. What could be the result of this? Okay, a mutation in a section of DNA, because remember, DNA codes for protein. A mutation in a section of DNA causes a ribosome to place an incorrect amino acid in a protein chain. Because remember, proteins are chains of amino acids. What could be the result? A, only the sequence of amino acids is affected, not any higher level of protein structure. Well, if you review that video on protein structure, you'll know that the sequence of amino acids, it actually determines the three-dimensional structure because of R group interactions. The sequence of amino acids will determine, where there are negative charges in the chain, where there are positive charges on the chain, where it's neutral, where it's polar, where there are covalent bonds that can form. So if you switch out a positively charged amino acid for a negatively one, you're going to completely change how that protein folds. So you can't affect primary structure without affecting tertiary structure and quaternary structure most of the time. Secondary structure is certainly affected because amino and carboxyl groups are different from one amino acid to the next. Maybe secondary structure could be affected by a mutation, but not for this reason, though, because carboxyl and amino acids, that's a repeating backbone. So that's the same from one amino acid to the next. It's the R groups that are different. Tertiary structure is likely to be affected because different amino acids have different R group properties. So changing an amino acid could change how the R groups interact with each other. Yes, this could be. Tertiary structure is likely to be affected because different amino acids have different R group properties. Some are positive, some are negative, some are neutral, some are polar. So changing an amino acid could change how the R groups interact with each other. Well, you know that opposites attract. So for example, if you have a positively charged amino acid in one part of the chain and a negatively charged amino acid in the other part of the chain, they're going to form an ionic bond with each other because opposites attract. But if you change a positively charged amino acid into a negatively charged one, they're no longer going to be attracted because like repels. So you're no longer going to be holding that loop together. It's going to be separated. So you know that if you change the sequence of amino acids in the chain, it has to change the tertiary structure. And then the last one, quaternary structure and tertiary structure would be affected because both structures are determined by R group chemistry. Yes, they are. Both are affected by R group chemistry. Different amino acids have different R group properties, so changing an amino acid could change the R group how the R groups interact with each other. Okay, match each image to the correct type of biological molecule. Hint, remember that unlabeled corners are always carbon. Okay, this one. Okay, here, we have carbohydrate, lipid, nucleic acid, protein. You have to identify, these are the four there's other biological molecules, there's vitamins and such. These are the four that we're covering. You have to be able to identify them. Okay, what's a carbohydrate? Well, it's built of sugars. So where's the sugar? Where's the sugar? All right. Well, the sugars we've looked at are six-sided. So that's a good hint right off the bat. And every corner that's unlabeled is a carbon. So 1, 2, 3, 4, 5, 6. 6 carbons. You know what the thing is? You can't, I get it. Like, you can't actually find the molecular formula in the shorthand. Maybe I should, because a lot of the hydroxyl groups are being left off here. I am expecting you to recognize that this is sugar based on the fact that it's a chain, these long chains. This is glycogen. We looked at these branching chains of glucose, which is a 6-sided sugar, 6 carbon and 6-sided. If that was confusing, I get it. Like you would look in here and you'd say, I don't know, what's a carb here, right? Because this one doesn't, sometimes biologists are just so lazy. I'm so sorry. Biologists are so lazy. They just get used to leaving things off. So maybe that's not the best one to start with. Maybe you want to leave that process of elimination. You'd be able to leave it off. Okay. This is glycogen though. Okay, so let's move on to lipid. Lipids are hydrocarbons. So here you don't have this is the steroid we looked at in class, and again, it's in the lipid structure. This is a cholesterol. So carbon, 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 carbon. These are all carbons, carbon, carbon, carbon. The only thing that's not carbon here is this little hydroxyl group. So this is the lipid. If you look at the lipids in the lecture video and then or in the PowerPoint, you'll see that we've covered this lipid. This is a cholesterol. It's the precursor for steroid hormones like testosterone, estrogen, progesterone, and also stress hormones like cortisol and things like that. So this is going to be your lipid. It is very hydrocarbony. Okay, nucleic acid. Nucleic acids have a sugar phosphate backbone and then nitrogenous bases that carry the DNA code. So here you have sugar phosphate. Sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, and this even has the A's, T's, G's, and C's, so the genetic code. So this should be immediately obvious that that's DNA or RNA. So that's nucleic acid. And protein has the repeating amino, carboxyl, amino, carboxyl, amino, carboxyl with the R group sticking out. So here's amino, carboxyl, amino, carboxyl, amino, carboxyl. Just going on and on and on and on and on. And then these R groups sticking off the side. Okay. So this should be immediately apparent as a protein because of the amino carboxyl repeating. This should be immediately obvious that it's a nucleic acid because of the sugar phosphate repeating and the A's, T's, G's, and C's of the genetic code. The lipid and carbohydrate in this might be a little harder given you're not, as exposed yet to how these things are drawn. This is just carbons, all carbons surrounded by hydrogens, okay? And then this one is left. It's got a lot of hydroxyl groups and oxygens. This is going to be your glucose chains, branching glucose chains for glycogen, which is a carbohydrate. Again, all of those organic molecules are covered in the online videos I've provided you with. Okay. Blank structure determines blank structure and blank structure of a protein because the sequence of blank acids determines how their blank groups will interact and fold up. Yeah, it's hard. I know this is hard, but I really want you to get this. So I've told you in the question that it's about protein, and you should immediately know that the acids in proteins that we're talking about are amino acids. So this is immediately amino acid, okay? Because amino acids are the repeating subunits in proteins. How they're blank groups. What are the blank groups? These are R groups. The structure of a protein is determined by R group interactions, okay? So again, if that is confusing, please rewatch the videos on the video on proteins. So blank structure determines blank structure and blank structure of a protein because the sequence of amino acids determines how their R groups will interact and fold up. I would just hope that these last two would be immediately obvious, and then the first two you'll have to figure out through hard thinking. Okay, so if these last two aren't immediately obvious, you really need to review those videos. Okay, so if we have blank structure determines blank structure and blank structure of a protein. Now, we've talked about different kinds of structures related to proteins. Primary structure, secondary structure, tertiary structure, quaternary structure. And this is just a repetition of a question that I asked earlier. A mutation in a section of DNA causes a ribosome to place an incorrect amino acid in a protein chain. What could be the result? So you have a change. This is talking about a change in primary structure because it's changing the amino acid sequence by changing which amino acids are there. And that's going to affect quaternary structure and tertiary structure. So I put the harder question after because if you answer this correctly, I was hoping that that would kind of give you the answer. Because you should know the answers here. So primary structure determines tertiary and quaternary structure of a protein because the sequence of amino acids determines how their R groups interact. And tertiary and quaternary structure depend upon R group interactions. How is protein shape related to function? Its shape dictates what other molecules are interested in it? No. Not really. So its shape dictates what other molecules can bind to and its shape dictates what other molecules it can bind to and what actions it can take upon those molecules. Yes. We talked about that lock and key thing and the oxytocin and all that. The cell won't recognize what it should use the protein for if it looks different than it once did. You should already know that this is wrong because I would never say looks different because these are not seeing molecules. I might talk about shape, but I'm not going to talk about how they look in terms of how other non-seeing things interact with it. Its shape dictates species attraction and compatibility. We're not talking about species here, so that doesn't even make sense. Okay, so its shape dictates what other molecules it can bind to and what actions it can take upon those molecules. That was sort of, yeah, so I covered that with the oxytocin and lock and key analogy. Changes in primary structure are highly likely to result in changes to tertiary and quaternary structure, change protein shape and protein function, preserve the shape of a protein, improve protein function. So if the primary structure of a protein changes, if the sequence of amino acids changes, is going to result in changes to tertiary structure for sure. There we go. It's also very likely to change the protein shape and function because the three-dimensional shape is determined by the sequence of amino acids. Not going to preserve anything if you're changing it. And improve protein function. You can't predict that. Maybe it'll improve the function, but more likely, if you just randomly change the shape of something that's been working, it's probably not going to work as well. That's just the most likely outcome. So that would be those two answers. Okay, I hope that that helped a little bit. I know that towards the end here, this was getting pretty difficult, right? So let's see, these are all 1 point. Some of these must be extra credit, I'm thinking. Let me see, I can't remember what I did here. Because each one of these is 10 points. Oh no, it's just partial points. Okay. Yep, so these, I know that like this one in particular, question 11, that's hard. It's really hard. I'm not going to deny that it's a hard question, but I really need you to be that familiar with these biological molecules for the exam and in order to move forward in the course, because we're going to build upon this after the exam. And so if you don't have this foundational material, the next component is just going to be super confusing. So this really has to be innate. It has to be intuitive to you by the time we move on. All right, let me know if you have any questions about any of that.